Namaste. I, JV and Dr. Chandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University. In today's class, we are going to discuss from the course Bioethics, okay, from which we will discuss about biosafety and its implementation. And okay, so when we talk about biosafety, means the need to protect human and animals health and environment from the possible adverse effect of the product of modern biotechnology. Okay? So the safety measures which prevent them from escaping from the harmful effects. Okay? So when we talk about this biosafety, the air filtration, sterilization of light, waste disposal, from the laboratory or the protective handling. So air filtration, the exhaust air from the laboratory is filtered through exhaust filter. It prevents escaping of the GEOs and from the labs. Then sterilization light, then fluorescence tubes which emit UV lights are fitted with the laboratory to sterilize the work area and expose the surface of the lab. This technique destroys microbial contamination inside the lab. Waste disposal, all waste coming out of the laboratory are sterilized by autoclaving or by uh, so that it become dead before throwing into the environment. Okay, it prevents escaping of the contamination waste from lab to the environment. Okay. Then protective handling. Person working in the laboratory must follow certain techniques to avoid contamination and to prevent escaping of the microbes. The person must wear protective clothes before entering the work area. It should be carried out. Mouth pipetting should be avoided. Biological contaminations like the principle behind this is to make if the contaminants make the organism unavailable to survive outside the fitted environment. It prevents the spreading of vector DNA to the organism outside the laboratory by using conjunction, transformation, and transcription. Okay. So when we talk about the bacteria which grows outside, unless a suitable nutrient have to be supplied for the gene manipulation. Such bacteria may include a gene mutation, and this mutation bacterium that survives only in the culture medium. Okay. So there are different levels of biosafety. Biosafety is the level of the biocontaminations, precautions required to the isolation or dangerous biological agents in an enclosed facility. It may be from lower to the highest level. Okay. When we talk about the biosafety level 1, it is suitable for the work involved well characterized agent, not okay. So when we talk about like a kind of bacteria and virus includes the canine or hepatite or non-pathogenic E. coli or some culture or non-infectious bacteria. Okay. When we talk about the level 2, it includes the uh, like moderate potential hazard to the personal and the environment, and it includes bacteria and virus that cause only mild diseases to human and are difficult to contact or via aerosol or in the lab setting like hepatitis A, B, C. The level is applicable to the clinical diagnosis. 3 is for diagnosed or teaching or research or production of facilitates in the work done. Okay, so it is a bit more or lethal diseases after inhaling the toxic gases or something, or bacteria or virus or pathogen may cause a fatal disease where the safety level 3 is maintained in a laboratory by wearing appropriate personal protective cloths and equipment. Okay. Then safety level 4, this is the level is required for the work with dangerous and exotic agent that pose a high level of risk in a aerosol transmitted laboratory infectious disease or the agent that causes severe to the fatal disease in a human. For vaccines and or other treatment are not able. Okay? So like Ebola virus and at present like coronavirus. Okay. So the biosafety level 4 has to maintain in this field when we are working with that in a laboratory condition or even in the field condition at present. This level is also used for work with the agent like where life risk is at the peak. So the biosafety guidelines aims at the regulating RDNA research from the organism that have least or no adverse effect, minimizing the possibilities of occasional release of GEOs from the lab, banning the release of GEOs from, if they are supposed to be the causing potential risk to the environment. Food storage, eating, drinking, or smoking are prohibited in the lab. Mouth pipetting is prohibited in order to follow the safeguard. Okay, laboratory coats are obligatory and should be removed when exiting the lab. So, if most of the time you see people use the coat in even though when they are out of the lab or a workplace, but it should be like the moment you exit from the lab, you should remove the your laboratory coats. Okay. So working surface must be decontaminated using soap, alcohol, or after each and every work every day. Waste products should be decontaminated by autoclaving it. Okay. Frequent hand washing is obligatory. Avoid contact with GMOs or other exotic biological agent disposed. Disposable gloves should be worn when handling such items. Laboratory doors should be closed at all the time. Work with fume producing chemical must be under the laboratory hoods. Biohazard warning signs should be always posted in the labs. So what are the risks associated for the environment? The susceptibility for the non-target organism, change in the use of the chemical in agriculture, unpredictable gene expression or a transgene or a gene slicing take place maybe, or risk weed or superweed can grow, alternative of the nutrient values can change, or change in the cost of the okay, different agriculture product may be changed. So there are a lot of ethic is associated with the risk, and social economic situations in a developing countries is also associated, so have to be taken care while working with the laboratory or lab when we are working with the any of the biological agents or organisms. Thank you. This session is powered by Digital Version 2.0 Jyoti Vidyapeeth Women's University, and hope you are satisfied with my digital session. If you have any queries, please mention in the comment box. I will resolve it.